And then this has the and the...
Okay, good. Okay, so I guess I'm charged with 10 years and 10 minutes. So I think it's, our project is or our lab is we do a lot of collaborations, and I direct an NIH-funded botanical research center. It's one of 5 in the U.S. It's an $8 million program, and we have 3 projects and 3 cores with it. And I'm just going to talk about that in these 7 or 8 minutes. So I think and then I'll talk about the project that I have in this. And a lot of this is actually what we do as cancer biology, but I don't really have a nanotechnology component to what we do. But I think as you look at each of the slides I show, you'll see that there's nanotechnology components to this. And I collaborate with JJ and even with Brian and others, so we have a lot of interactions. So the overall goal of this Botanical Research Center is to, the title of it is Botanical Estrogens, Dose, Timing, and Target Tissue. Probably the most widely used dietary supplement are those that are targeted to women over 50, and there's no shortages of these things on the marketplace. And what we wanted to do with this Botanical Center is to provide basic information on how these estrogens work and characterize their mechanisms of action. And then we want to examine their effects in important target tissues. And the target tissues we're looking at are going to be uterus, mammary gland, bone, and brain, and breast metastasis. And the ideal here is that we want to offer at least a conceptual framework that gives a science base as to how these work. I mean, there's no shortages of claims as you look in the third-party literature associated with these dietary supplements. The ones that we have focused on are ones that I would call that are in the top 10 of those being consumed by women. And that's going to be the soy isoflavones, and then the metabolite equal, wild yam, licorice root, and dong quai. And we're mostly going to we'll probably run out of time before we get to this one. So the other 3 projects, the first project is one that's led by Benita Katzenellenbogen, and her husband is a co-I on this, and it's a combination of a molecular biologist as well as a chemist. And this is the molecular basis project. And what they will do is examine botanical estrogens and then see how they bind to ER alpha and ER beta, and then look at patterns of ligand binding and co-regulatory interactions. And a very simple way to put this is, there's no shortage of these products in plants. And the products that women are consuming in general are going to be extracts. And we need some way to be able to get an idea of whether these extracts have some action in a cell system. And so the first project actually will help us identify which products to identify or further study in vivo models. And then also, once we have in vivo models, then bring it back to this project where you can actually look at the mechanisms by which these products may work. The second project is the project that I lead, and this is looking at the actions of botanical estrogens in bone, uterus, and the area that we have been working on over the last 5 years is looking at ways that alter metastatic progression. So, I mean, we have collaborators at Oregon State that actually do the bone work for us. And so we'll evaluate these botanical estrogens. And early on, we're going to use uterus and mammary gland. These are things that we can do in a short period of time in an in vivo model. And then we'll look at metastatic progression. In late-stage breast cancer, 70% of these women have metastasis in the bone. So it becomes a real critical issue as to what kind of dietary interventions, whether it's a supplement or a nutrition component, that can actually alter the progression of this disease. And it's interesting, but we're finding that just simple dietary changes can actually change the metastatic progression. So, and then we use in this project we use bioluminescence imaging, and I have 2 slides at the end of this that'll talk a little bit about what we do specifically. The third project is one that looks at botanical estrogens and cognition. This is led by Sue Shantz in Vet Med, and her project is almost all preclinical model evaluations, and she does cognition evaluation to see whether the animals enhance or reduce their ability to with regard to their cognition. And then we have 3 cores that are associated with the the center. One of them is an administrative core, and I won't spend much time on that, but the first one is identification and characterization and quality assurance. Again, just like there's no shortage of these products, there's no shortage of adulterated products. I mean, we purchase things from China, and when we get them to the U.S., they're adulterated. I heard at a meeting last year that about 40% of the products that are sold, the FDA has identified those as not complying with the regulations, meaning that they could be adulterated, they could have more than what they say they have in them, or not what they say they have in them. So I think this is a real critical core, and it's something that NIH really requires that we confirm that what we say we're working with is actually what we're working with, and confirm that we have batch-to-batch similarities. We have a pharmacokinetics core, and again, as you can imagine, dealing with these extracts, you need to have some mechanism to know how much you're administering. So once we identify the bioactive that's present in the extracts, then we do pharmacokinetic modeling of that to be able to determine what's in the animal, what's in the tissue, and then use that to predict what may be in the humans. And then just the overall structure of this grant is, again, we have the administrative core, the three projects I just described lead into it, and then the characterization core and the pharmacokinetic core all support these projects. And we just finished the second year of the 3-year projects, and we're hopefully they will do another round of these grants, and hopefully we'll be competitive at renewing this grant. So what I'd like to do is just in the last 2 or 3 minutes that I have, I'll talk a little bit of detail about what we actually are doing. So what we're doing is we use bioluminescence imaging to develop a preclinical model of metastasis. 10 years ago, it was very difficult to do metastatic studies because you wouldn't know when to harvest the animals to see where the tumors would be or where they have progressed to. So what now we're able to do is to follow in real time metastatic progression. So what we use is a tumor cell, a mammary tumor cell that's been engineered to overexpress luciferase. And so these cells will actually express light when the animals are injected with luciferin. So in this case, now, as I mentioned earlier, that in late-stage disease, most 70% have mets in the bone. So we actually mimic a metastatic tumor in the bone by putting a few hundred cells into the bone, and then after a week or so, these tumors grow fairly aggressive in the bone, and then after several weeks, they will actually metastasize from bone to visceral tissue. In this case, it's usually lung. So we're allowed to actually, in this model, we can actually follow the tumors in real time, progressing from primary site to a tertiary site. And what's nice about this is we know when to harvest the animals, so that when we do the lion's share of the work in terms of histopathology, we know what tissues to look at and when to collect the animals. This is an example. This is a leg of a mouse injected with saline, and then this is what these tumor tissues do to the bone. They're very, very aggressive in terms of causing remodeling of the bone. So with that, I wanted to keep this short because there probably will be some questions. We've been pretty well supported to do this over the past decade and a half, and I've just listed where some of the money comes from. Most of these are current grants. So are there questions? I think I've got 2 minutes. Probably, I think I took your 10 minutes pretty literally. So. Go ahead, Rashid.
What's the spatial resolution in that imaging in the bioluminescence? Can you look at CPCs?
No, the resolution with luminescence is really quite poor. I mean, I think what Steve Bopart's doing is he's using the same instrument, but he's actually got resolution that's a lot higher. What our goal in this is just to identify that the tumors have actually progressed from one site to another. And then that allows us to either have cohorts of animals that are just beginning to cause metastasis, or to follow the metastasis. And because if you're trying to get an idea of ways that can be to alter the progression, you can't have the animals go too far where you can't detect any differences. So we just use it as a tool. It's not very sensitive, and everybody likes to think the companies call it three-dimensional, but it's not three-dimensional. If this was, say, the liver and the tumor was here, and you're imaging from here, it's going to be a very bright tumor, even though it may be small. Then you could have a large tumor under here, and you wouldn't get good imaging on it. It's just a nice tool to be able to follow the tumor progression in real time. Other questions? Okay. Oh, good. Hope that
Thanks so much.
Sure.
Great, thank you.